Hello all, welcome back to the BioDocs channel. In this video today we are going to learn about the ABO blood groups or the ABO blood group typings as well as about the RH antigen also. Okay, so our blood groups, actually the classification of the present day ABO blood group systems was being given by Mr. Carl Landsteiner. And our blood groups are named based on based on the type of based on the type of surface antigen surface antigen present on our RBC membrane. Okay, so this is important to remember. So based on what type of surface antigen is present on our RBC membrane, we give that sort of name to the blood group. For instance, if I'll give you an example, if an RBC membrane, a person's RBC is going to comprise of antigen, which is being called as A. So for the convenience, I'm just writing it as A, but it is not this. I'll explain you what are these. If they have the surface antigens as A, then the let me write it. If the surface antigen present here in the person on his RBC membrane is going to be A, then the blood group that we are going to classify here is going to come under a group called as blood group A. And if the person is going to comprise the antigen B on his RBC membrane, so the surface antigen is going to be B, then the person's blood group is determined as blood group B. And if a person is going to comprise of both the A as well as B sorts of antigens on their surface, so if it's going to be A as well as B, then we are going to call such a blood group as AB. And if a person's RBC membrane do not comprise of either A, there is no presence of A nor B. Okay, so then we are going to call such blood group as blood group O once again. So based on the antigens present on the surface, we are calling or we are classifying them into such particular group. So if they have a A antigen, then they're under the A blood group A. And if they have an antigen B, then it is going to be the B blood group. If they have both the antigens A as well as B on the surfaces, we call them as blood group AB persons. And if their RBC do not comprise of no antigen A nor no antibody B, sorry, antigen B, no antigens, okay? So they are not comprising of the antigens A as well as B. So they have an absence of antigens of these two, that is A and B, then we are going to call it as a O blood group we are going to call, okay? But we have to know now that what are these antigens, okay? So the surface antigens actually are, they are the surface markers. They are the surface markers, okay? And these antigens actually are responsible for immunological responses, immunological responses, okay, responses. The meaning of this is going to be that they are going to actually they trigger the antibodies, due to which the antibodies are going to act against the respective antigen, okay, if they match. Okay, so if the antigen as well as the antibody match, then this is going to lead to clumping of the blood, or we call this as agglutination. We call this process as agglutination. And during the clumping of the blood, what happens is this results in hemolysis. hemolysis of your RBCs, which is nothing but your RBCs are going to get ruptured. Okay, so this is going to lead to the ruptured RBCs. So this is dangerous. So before the blood transfusions, we have to check the blood types. Okay, blood types. So what are antigens? Are nothing but antigens are a type of glycoproteins which are present as surface markers on your RBC membranes. Okay, RBC membranes. Now, children, now what I'll do is I'll take you through the journey of these glycoproteins. Let me show you a few slides. But before that, just remember that your blood groups are determined. Blood groups are determined based on based on the antigen on your RBC membrane. But remember, children, to actually the blood groups, antigens are nothing but just I spoke about, they are going to be the sugars, because I told you they are the glycoproteins, and they are actually the sugars which are clubbed up with the proteins in on your lipoproteinaceous membrane, but they are basically the sugars. Antigens are the sugars. So in order to synthesize these sugars, see, in order to synthesize these sugars on your RBC membrane, what you require is you are going to require for this, we are going to require some enzymes. We require some enzymes as well as, and this is going to be as well as we require some proteins also. But the basic most important point I need to make you understand is your RBCs do not have nucleus. If they do not have nucleus, they do not have they do not have any genes. They do not have any genes, nor they do not have any enzymes. They do not have any RNA or ribosomes to actually synthesize. So they do not synthesize these sugars. Actually, they do not synthesize sugars, or they are unable to. So my question is, from where are we getting the sugars? Where are we getting sugars on the RBC membrane? So this is very important. And the answer for this is going to be actually, it is the precursors of your RBCs. Before the formation of the RBCs, there are some precursors, or we comprise of some erythroblast cells. We have RBC cells. Actually, they are the reticulocytes, what we are going to call them as reticulocytes. We have, so these are the ones which are the precursors of RBCs, and these precursors of the RBCs, nucleus is present in them, but only when they form as red blood cells, the nucleus is being lacking. Okay, so nucleus is present, and they will even have the required enzymes and sugars to make required enzymes and proteins to make your sugars on the RBC membrane. This is very important to remember, that the mature RBCs do not contain a nucleus, but it is precursors from where the RBCs form. They do have a nucleus, which are going to have enzymes as well as the proteins which make these sugars on your RBC membranes, children. So remember that there are two chromosomes actually, the chromosome number 19 as well as the chromosome number 9. Okay, the chromosome number 19 is very important in adding a sugar to the this particular RBC membrane, that is fucose, and chromosome 9 is going to add up the sugars what you require to make, so they are going to make the sugars that are required to make your blood group A, blood group B, okay, and the blood group A, B, that we'll see now. Let me share those slides with you, children, such that you will understand now that what are those which are going to actually help in making the different blood groups what we are trying to learn. So this will give you a basic idea regarding that. So children, let me introduce you with this particular slide. Okay, first we are going towards one particular antigen. So let me show you what is this. This is your RBC, and on the membrane of this RBC, you are going to have a basic oligosaccharide. What is an oligosaccharide? Is nothing but a group of simple sugars forming a chain like this. Okay, they're forming a chain like this. This is going to be the protein part, and these are going to be the sugars which are being present, which is going to be a basic oligosaccharide made of glucose, galactose, N-acetyl glucosamine, and galactose. So this is not in your portion, but I just wanted to clear the topic of what are antigens, because such that you will understand what are those glycoproteins. So there's a protein with the chain of the sugars, which is being called as oligosaccharides, children. So these all are going to be the oligosaccharides. So let me write this for you, children. So these all things that you're seeing from here to here is going to be the oligosaccharide. So now you can see that the oligosaccharides which are being present here are the basic things, but the chromosome 19 which is present, the chromosome 19 is going to actually make an enzyme. It makes an enzyme which is going to transfer this sugar, which is being called as fucose, to this common oligosaccharide present. This is a glycoprotein present, and you are getting an addition of a new sugar, which is being called as a fucose here. Okay, this is a simple basic glycoprotein, which is being called as H antigen, which is being seen in the blood group O. This is seen in the blood group O, because I told you that in the blood group O, there are no antigens like A and B, but yet they have an antigen which is being called as H antigen. That is not in your syllabi, but to make you understand that this is a basic structure of the oligosaccharide with the sugar added up, which is being called as fucose. So these are the sugars which are making your blood groups, which are responsible for giving you the blood groups. Okay, and the next thing that I would like to show you is going to be the another antigen. So this is going to be the formation of A antigen, which is responsible for the blood group A. You can see that this is a common oligosaccharide chain that we have here, and we already have seen that the chromosome 19, let me please give me a moment for this. So we are going to have a chromosome 19 coming from the parents. So the chromosome 19 here is responsible for the production of an enzyme, which is responsible for transferring this particular what we call is sugar here, and this sugar is fucose, which makes this basic structure is being called as H antigen, which is being seen in the blood group O. Okay, but now what is happening is the chromosome 9 now is going to produce an enzyme. It's going to give up an enzyme which is going to transfer a sugar which is being called as N-acetyl galactose amine. That is this particular sugar, due to which now we are seeing that the basic H antigen which is being present, this is the basic prime H antigen to which a sugar like N-acetyl galactose amine is attached, which makes up the blood group, which is being called as blood group A. And now this whole structure now with the fucose and the N-acetyl galactosamine is going to make the antigen A. So this is going to be your antigen A. I hope you are understanding that how sugars attached to this particular oligosaccharides are making up your what we call as the antigens and the RBCs, which are responsible for the formation of the different types of blood groups in the human beings and the other animals. So you can see now that the formation of the B antigen we are speaking about. So when I speak about the B antigen, what happens is you can see that the basic idea is again that the chromosome 19 is going to add up, produce an enzyme which is going to transfer the fucose sugar, which makes a basic H antigen, which is present in your blood group O, but they do not have blood group A and B. That is antigens A and B they do not have. The reason is, now you can see the magic that the chromosome 9 is making an enzyme which is actually adding up a different sugar here to the H antigen, the basic oligosaccharide branch here, and that is none other than your galactose sugar. And because of addition of the galactose sugar to the antigen, so you are adding a galactose sugar. So since you're adding a galactose sugar to the H antigen, now it is making up, this is going to result in the formation of B antigen. And as you know that whichever the antigen is present on the membranes of the RBCs, you get such type of a blood group, and this is seen in the blood group B persons, this particular RBCs. Okay, children, I hope you are understanding what I'm trying to explain you. I wanted to show you about those glycoproteins, the addition of the sugars and the variations of the sugars, which are going to make up actually the basic difference between what is blood group A, blood group B, blood group O. But remember, children, if a person is going to have, if his chromosome, let me please give me a moment for this. So if a person is going to actually have both the A and B, so now what happens is a person's chromosome 9, that is coming from a maternal and paternal, what they are going to have is both are dominant in producing their own respective enzymes. So the maternal chromosome producing the what you call is enzyme, which is going to transfer this particular N-acetyl galactose amine, as well as it is going to, the paternal chromosome is going to transfer the galactose. Then it is going to make a B antigen. So here you can see now the change is, along with this, they are going to have another sugar which is added up here, which is going to be your N-acetyl galactose amine, which is going to make the A, and this is going to make the B. So they're having, along with the H antigen, they are having the new two sugars. One is responsible for the B blood group, one is responsible for the A blood group. So they are having both the A and B antigens. If this is going to happen, A and B antigens, then we are going to call such a person as blood group A, B, we are going to call us. So I hope, children, this has given you a clear idea regarding like what are those sugars, which are actually responsible for making up your blood group. So I hope, children, you have understood that blood group O person is actually not devoid of any antigens, but he is not comprising of neither the A nor the B, but he has a basic antigen, which is going to be the H antigen. This is actually the thing which is running on. Okay, so this is a basic idea regarding the blood groups that we need to understand. But remember, children, I told you that antigens are these sugars which are present on your RBC membrane, which are immunologically very active. Okay, they are immunologically very, very active. They are going to trigger your antibodies. Okay, and they have antigen antibody reactions, which if they are matching up, it's going to lead to clumping, clotting, agglutination process. Okay, but where are the antibodies present? I haven't spoke about the antibodies. Antibodies are going to be the proteins produced by your WBCs. Okay, proteins produced by your WBCs, and these antibodies are going to be like, you can see that the blood group A, let me take these. Okay, blood group, now I'm speaking about A. So the antigens present here are the antigen A, so we call it as blood group A. But naturally, these antibodies are naturally occurring. These are naturally occurring, and luckily we see that we do not have an matching antibody, but we have another irrelevant antibody which does not actually react with the antigen A, and that is going to be your antibody B. This is, we are going to have, these are present in your blood plasma. Okay, these are present on your RBC membrane, these are present in the blood plasma, and these antibodies can be regarded as anti-B. And blood group B, now we'll be considering the blood group B. As we know that antigen B is present, so we call it as a blood group B, and they have an antibody A, and we have blood group AB, which is going to comprise both the antigens, that is going to be A as well as B. We have seen a while ago, and they do not have any antibodies, because if they would have had any of the antibodies, they would have reacted with these antigens because they're very immunologically active. They will trigger the antibodies, which will lead to the agglutination and hemolyzing of the RBCs, which is going to lead to the death because of the blood clot. And the next thing that we're going to see is going to be about the blood group O, which is going to be actually does not comprise, it does not have any, no, so it's going to have no antigen A, nor it does not comprise an antigen B at all here. And they do have antibodies because they don't have the antigens, so they have both the antibody A as well as they're going to have antibody B. I hope children you understood now. Now let me speak about the rhesus factor, which was being first time seen and discovered in rhesus monkey. Most of the 93% of the population do possess this particular protein, the antigen, which is being called as rhesus. So now we will try to see what happens is, if a person, I'm drawing a person with blood group A, I'll take an example here. So this is going to be the blood group A person. And remember children, rhesus factors are, there are three subfactors actually, but amongst them, if you're going to see the rhesus D factor subfactor is most predominant. So predominant, and we consider this rhesus D the most in most of the population. As I told you, it's been seen in the 93% of the human population, that we are going to have these what we call as the RH type of blood groupings we have. And if you're going to see, we even have the RHC as well as the rhesus C subfactor we have, and we even have the rhesus E factor, but this is most predominant and we are considering this. So if this is going to be blood group A, it is going to have an antigen A, as I told you, but this is also going to comprise of antigen, which is going to be the RH antigen. So now this blood group A is being called as A positive. Okay, now let me take another example here. If I will take an RBC with A antigens over the plasma, that is your membrane here, but if they do not have any RH here, then I should consider this particular blood group as a blood group A negative. We are going to call it as blood group A negative. Same case with all the other blood groups. Let me consider as O now. I do not have any antigens, but it is comprising of RH then this particular blood group should be considered as O positive. And if I'm going to have an RBC with no antigens and no RH, then I should consider this particular individual as O negative, and the universal donor is going to be this O negative person. Children, I'll be making a special video to check who can actually donate, who can actually receive blood from the different blood groups. I'll be making a special video on that. Please do give a look at that particular video, because that is going to actually help you in understanding the procedure, how to determine how which particular blood group is going to be actually responsible for giving the blood or donating the blood or taking up the blood from other blood groups. But here to remember is, the blood group AB, which has been present here, is being called as a universal recipient. It's been called as a universal recipient. It can receive blood from all the other blood groups, and the blood group O is being considered to be as a universal donor. But when we consider the RH factor, I told you that it is going to be your O negative persons who are being called as a, what we are going to call as a universal donor, we are going to call them as. So these are few things that you need to remember, children. And basically speaking about two terminologies, antigens can otherwise be called as a agglutinogen. We are going to call them as an agglutinogen. And antibodies are being called as agglutinin. And you know that when they are going to react, if they are matching each other, that is going to result in the agglutination process. So when an agglutinogen, that is an antigen, is going to match with your antibody, which is being called as agglutinin, and if this is going to lead to clumping, then this is going to lead to the hemolysis of your RBCs, which is going to be called as agglutination process, which is not a yeah, what you call is a process that has to be happen. So we have to check the blood types before the, what we call as blood transfusions, because if they are going to, if the antigens are going to match up the antibodies, that will be leading. For example, if this is going to be antigen A present, and if this is going to match up, that is seen in the blood group B, like antibody A, then this is going to lead to, see, this is A and A, so they are going to lock up with each other. So that is going to lead to the agglutination process. So these are the things that I needed to speak about. What is RH factor? And that is also an antigen, children, just remember. So this is, we are speaking about the antigen RH that is being seen in the rhesus monkey. And speaking about the chromosomes and the sugars which are making up. So I hope you understood it clearly, that what makes up your blood groups. Okay. So if you like the video, please do hit the like button. In the forthcoming videos, I'll be making about the videos regarding the blood transfusions. I'll be working on who can actually donate, who can actually receive. I may give you a few of the examples, and we will even work in the other videos on the genetics of the blood groups also. I hope you have understood, children. Please do subscribe my channel, as well as try to share my channel with others. That is going to help them a lot. And yes, thank you all. Thank you.